Hi guys, welcome back to the channel. In this video I will be going through the structure of DNA and RNA topic of the A-level biology curriculum. Let's begin. So first you need to know what nucleotides are. So nucleotides are basically monomers of nucleic acids. So lots of nucleotides join together to form the polymer nucleic acids, and the nucleotides are made of phosphate groups. So all of the nucleotides have some things in common. These include the phosphate group, the organic base, which is also sometimes called the nitrogenous base, and a pentose sugar. So looking at the diagram for a nucleotide, so as you can see, they always have this phosphate group, and they have the pentose sugar, and also the organic base. So it is the pentose sugar which is going to bind to both the phosphate group and the organic base, the nitrogen containing base. So the nitrogen containing base will be on the right, attached to this carbon, and the phosphate group will be on the left, attached to this carbon. And make sure when you're drawing this you don't draw the line from the middle, because the corners there suggest that this is a carbon, but the middle would not suggest that this is a carbon. So make sure that you draw from these corners, these edges. So next, a condensation reaction between two nucleotides forms a phosphodiester bond. So for example, if we bring along another another nucleotide, what happens is the phosphate group of this nucleotide would attach with the pentose sugar on the other nucleotide, and we would basically form a phosphodiester bond. So this would form a phosphodiester bond. So a condensation reaction between two nucleotides forms a phosphodiester bond, and we would also have a release of a H2O molecule, as this is a condensation reaction. So next, focusing on DNA now. So DNA is also called deoxyribose nucleic acid. Make sure you know the full name. And this is, again, it's a polymer of nucleotides. So because it's a nucleic acids are monomers, are polymers, sorry, and the monomers would be a nucleotide. So DNA, what it does is it holds the genetic information in all living things. So the genetic information is held by DNA, and all DNA would contain a deoxyribose sugar. So before we mentioned that all nucleotides have a sugar, but DNA in particular would have a deoxyribose sugar. This is why it's called deoxyribose nucleic acid. And it would also have a phosphate group and the organic base, so it can only have one of the four organic bases in one of the DNA molecules. It can only have either adenine, cytosine, guanine, or thymine, so one of these. Now you will find out this is slightly different to RNA when we move on to RNA, but this would be the structure of DNA. So as you can see, the phosphate group, and this would be the deoxyribose sugar and the base, so adenine, cytosine, guanine, or thymine. And there is something known as base pairing. So when we have the inorganic bases containing the nitrogen, so what happens is that base can pair up with another base from another nucleotide, and the base pairing rule is that adenine always goes with thymine, so A to T, and cytosine always goes with guanine, so C to G. And the DNA has strong phosphodiester bonds between the phosphate and the deoxyribose sugar, and this creates a sugar phosphate backbone. So as you can see in the picture, so when all of these the phosphodiester bonds are created, they line up to form this sugar phosphate backbone, which is quite strong. So the DNA molecule is a double helix with two polynucleotide chains. So as you can see that this would be one chain and there would be another chain which is paired up with this. And the chains are held together by hydrogen bonds, and these hydrogen bonds are between the specific complementary base pairs. So when we have the A to T and the C to G, the complementary base pairs are joined by hydrogen bonds. So as you can see, for example, in the first example, we have A to T. So this would be joined by these hydrogen bonds, and there are many hydrogen bonds which pair up the two DNA strand, making it a double helix. So double helix basically means there are two strands. Okay, so the structure of DNA and its function. So as the DNA is double stranded, as it has two strands, what this means is that both strands can act as templates in semi-conservative replication. So we'll go on to replication after, but what this means is because the DNA has two strands, so there are two strands, it's double stranded. So but when both of these strands separate, what this means is both of these strands can act as template in the semi-conservative replication, so when DNA replicates. And because DNA has weak hydrogen bonds between bases, so even though it has loads of these hydrogen bonds, but because they're quite weak, they are nowhere near as strong as the covalent bonds, and they can be broken easily. So the DNA, that's why DNA can be easily unzipped. So when these hydrogen bonds are broken, the DNA can be easily unzipped for replication. And because it has many hydrogen bonds, so every base pair would have a hydrogen bond, so this gives DNA stability. And because there is complementary base pairing, A to T, so adenine to thymine, C to G, cytosine to guanine, this allows the DNA to have accurate replication. So as the DNA is double stranded, it's double helix with sugar phosphate backbone. So this was the sugar phosphate backbone, so this carrying on for the whole DNA molecule. So this basically protects the bases which will be inside that. So the double helix is basically outside and the bases will be inside. So this protects the bases and it provides strength. And because DNA is a very long molecule, much longer than RNA, it can store lots of information. That's why it's useful. And because it's a double helix, it's double stranded, and this means it's compact, so a lot of information can be stored within a small space, so it does not take up a lot of space as it's compact. So RNA, so moving on to RNA, so now moving on to RNA, so RNA is called ribonucleic acid. So when we had DNA being called deoxyribonucleic acid, RNA is ribonucleic acid, and again it's a polymer of nucleotides. So this time it's single stranded, so where DNA was double stranded, this is single stranded. So comparing this is DNA with its double strands, RNA only has one strand, therefore it's single stranded. And RNA transfer the genetic information from DNA to ribosome. So that's what it does. That's the job of RNA. Next, the ribosomes are formed from RNA and protein. So this is one of the mark points of this topic. So you need to know that RNA and protein are required to form the ribosomes. And so the RNA, what it contains is that it has a ribose sugar. So compared with DNA having that deoxyribose sugar, it has ribose sugar. It has again the phosphate group. So the same with every nucleotide, it has that phosphate group. And it has one of the organic bases, adenine, cytosine, guanine, or uracil. So DNA had that thymine, but instead of having thymine, RNA has uracil. So again, looking at the structure of RNA, So as you can see, it has the exact same structure as DNA, but instead it has ribose sugar, and in that base, instead of having thymine, it has uracil now. Okay, so now the base pairing, so the DNA had that A to T and C to G, but RNA has that A to U base pairing and C to G. So adenine to uracil and cytosine to guanine. So there are a lot of exam questions that come up, and they just ask you about the differences in DNA and RNA. So here is basically a summary of them. So DNA has deoxyribose sugar, whereas RNA has ribose sugar. DNA has the base thymine, whereas RNA has the base uracil. DNA is double-stranded, RNA is single-stranded, as you can see in that picture, and DNA is much longer than RNA. So RNA is shorter than DNA. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.